Hello, everyone, to today's edition of the Polyploidy Webinar. Today we have two great talks. Up first is Justin Conover, who is currently a PhD candidate in the Interdepartmental Genetics and Genomics program at Iowa State University, where he works with Jonathan Wendell studying various aspects of molecular evolution following polyploidy and cotton. Justin grew up in a small farming town in northwestern Missouri, and in 2015 he graduated from Missouri State University with his Bachelor's of Science degree in biology. And Justin's going to tell us about the work that he's been doing since he started his PhD just a few years ago, it seems like, in Jonathan's lab. And he'll tell us today about his work on mutation load in cotton, the impacts of polyploidy, domestication, and speciation. With that, I'll let Jonathan take it away, or Justin take it away.
Don't worry, you're not the first person to get us confused. Thank you, Mike, for that kind introduction. Yeah, as Mike said, I'll be talking to you guys today about mutation load and how polyploidy, domestication, and speciation can all impact how much load is in a given species. Before I get started, though, I just want to thank the members of the Wendell Lab, who are all shown here, as well as Josh Udall at the USDA, who led a big resequencing project, which is where I got my data. And then I've also been funded through NSF and Cotton Incorporated through a lab with Colorado State University as well. So whenever people think of population genetics, they normally think of how to detect positive selection. But another aspect is how to detect purifying selection as well, because there's deleterious mutations that are in every genome, and selection is constantly acting on those mutations to purge them out and improve the fitness of the population. This has been fairly well studied theoretically, so we know that there's a lot of factors that can influence how deleterious mutations accumulate. One of the most prominent ones is the effective population size, where the larger the population size, the more efficient that population is at purging out deleterious mutations. Recombination also plays a huge role. If you have regions where there's not very much recombination, you can accumulate deleterious mutations quite frequently, whereas if you can pair them up through recombination, it's easier to purge multiple deleterious mutations at a time. Also, life history has an important part in deleterious mutation accumulation. So we know that asexual individuals tend to accumulate deleterious mutations more often than sexual individuals do. And finally, genetic dominance also plays a role here. So if there's a mutation that's, say, lethal and recessive, it can still segregate in the population at really low frequencies, but as soon as it's seen as a diploid or in a homozygous state, that individual will be purged from the population. One aspect that hasn't really well been studied in deleterious mutation accumulation is the impact of gene and genome duplication. So if anybody's familiar with population genetics, they've probably heard of the name J.B.S. Haldane. And when he was developing his basic theories of population genetics, he actually thought quite a bit about polyploids. And in his book, The Causes of Evolution, he was thinking about the impact that epistasis could have on relaxing, purifying selection. And he wrote what's become a fundamental theory in polyploid evolution, which is that if one gene can retain its function, its homolog can be mutated and can be changed without disadvantage. And this sort of logic has been used to infer, the causes of neofunctionalization, subfunctionalization. But as far as a population genetics approach goes, this has not been very well tested at all. There are a few ways you can detect deleterious mutations. The method I'll be talking about today is called genomic evolutionary rate profiling, which is a comparative genomic approach. So essentially the way that these approaches work is you create a multi-species whole genome alignment, and you estimate the rate of change at every position in the genome. If you use synonymous sites, where it doesn't matter which nucleotide you change it to, you always get the same amino acid. Those are considered fourfold degenerate sites. And that using those sites can give you kind of a basal rate of evolutionary rate among all these species. So if there was no selection acting at those sites, they should be evolving at the same rate as these fourfold degenerate sites. In the same vein, if there's a site that has less variation than you see at the neutral evolving sites, then that's inferred to be somewhat deleterious because there's been some sort of purifying selection that's resisted that position in the genome changing as fast as the neutral rates do overall in the genome. This is a really powerful method because it can be used for any gene and genome sequence as long as you can create a whole genome alignment for it. And specifically for this method, you don't have to rely on coding regions. You can also apply on non-coding regions. There are other methods that are based solely on the gene sequences, but this method allows you to look at, say, regulatory regions upstream of a promoter region as well. For this study, probably unsurprising if you heard Jonathan Wendell's name mentioned, we'll be using Gossypium, which is a genus of about 45 diploid species that are scattered all throughout the world. They're divided into 8 different subgenome groups, which are denoted A through G and K. For the important ones, D over in the Americas is a diploid ancestor to one of the polyploids, and the A subgenome is the other diploid progenitor to the polyploids. So we know that the A and D diploid genome groups diverged approximately 5 million years ago, and somewhere around 1 million years ago, there was a hybridization event followed by whole genome duplication, which has since led to the diversification of what's now 7 currently recognized polyploid Gossypium species. They're found mostly in the Americas, with Gossypium hirsutum, or 81, being the species that most of our cloth is made out of. But Gossypium barbadense, or 82, is another domesticated species that's the more expensive cottons as well. There's also been a few interoceanic dispersal events. So 83 is found only in Hawaii, Gossypium tomentosum, and Gossypium stephensi 87 is found in the Wake Atoll. The Wake Atoll is a really, really small Air Force base in the middle of the Pacific Ocean. And this species was only recognized in 2017. So for simplicity, so you guys don't have to remember the species names, I'll just be referring to these polyploids by their AD and then the number for that. So the data set that I'll be using is a big resequencing project that was led by Josh Udall at the USDA, and a lot of the analyses were done by Daojun Wan, who's now in China, but he was in Jonathan's lab as a postdoc for a while. And in total, we sequenced 643 accessions of all 7 polyploid species. So for this project, I sampled 40 individuals of AD1, which includes 10 wild individuals, 10 domesticated individuals, and 20 individuals from land race populations that are somewhere in between wild and domesticated. For AD2, I had 30 individuals, 10 that were domesticated and 20 that were in land race populations. Unfortunately, there's only a couple of wild accessions that anybody's found of AD2, but I didn't include them here because the sample size wasn't large enough. And then I've sampled various numbers of AD3 through 7, and then 4 individuals from D5, which is the paternal diploid progenitor, and then 14 and 10 accessions from A1 and A2, which are both equally distant to the A subgenome progenitor. For these methods, I also use some Australian cottons to polarize the SNP calling so we can actually tell what's the derived SNP and what's the ancestral SNP, which really helps elucidate some of the patterns that I'm seeing in some of this data. So first, I want to look at just the deleterious SNPs that are segregating within the polyploids or the diploids, but happen to be fixed within a specific species. So here on this plot, we're looking at the A genome of A1. The light green indicates mildly deleterious mutations. The kind of darker green is the moderately deleterious mutations, and the dark green is the highly deleterious mutations. This is based on the group scores as well. And we see a fairly appreciable number of deleterious mutations that are fixed in A1. When we look at the subgenome of 6 wild polyploid populations, we see a really interesting pattern. We don't see that there's always more fixed deleterious mutations in the polyploid than the diploid. There's not always less in the polyploid and the diploid, and sometimes they're all pretty close, which I think is an exciting trend and kind of counterintuitive to what I would have expected to see. If we shift our focus to the D subgenome now, to the D diploid, we see a lot more deleterious mutations that are fixed within the D diploid. And again, when we look at the polyploids, there's some species that have fewer deleterious mutations in the D subgenome than in the diploid, and some species that have more fixed deleterious mutations than in the D diploid. But what's really interesting in this pattern is that when we actually compare between the subgenomes, we see that generally, in every case, there's more fixed deleterious mutations in the D subgenome than in the A subgenome. And this pattern hasn't been observed before, which leads to a lot of questions, like why would there be more mutations fixed in the D subgenome than the A subgenome? And is this just due to effective population sizes? Is it a mutational difference between the two subgenomes? Is it differential introgression? Those are all open possibilities, and we're still not sure what the cause of that could be. One possible cause is this is just an artifact of the data, because the D diploid progenitor is a worse model than of the DT subgenome than the A diploid is of the AT subgenome. So if we look on this red part of the tree here, that branch length is different for the A subgenome and the D subgenome. So I actually subsetted the data again and took out any deleterious mutations that were fixed only in the polyploids and any mutation that was fixed only in the diploids. And the pattern still arises here, where generally there's more mutations that are fixed in the D subgenome than in the A subgenome, and there's still variable numbers when you compare the polyploids to the diploid. Two of these samples kind of stood out to me, 83 and 87, as having kind of higher than average deleterious mutations. And if you remember from that figure, these are actually the two species that are found in islands. Because this was probably a really long transoceanic journey, there's probably a very big genetic bottleneck for that founder event. And so my very preliminary and rough hypothesis is that because of that founder event, there was a large amount of deleterious mutations that became fixed in that population almost immediately upon colonization of those islands. Another great benefit of the Gossypium system is that there's been 4 different species that have been domesticated. 2 of them are diploids that were domesticated in Africa, and 2 of them are polyploids that were domesticated in the Americas. 3 of those species are shown here: Gossypium hirsutum 81, Gossypium barbadense 82, and Gossypium herbaceum A1. In the photo here on the left-hand side, you can see what the wild cotton seed looks like, and on the right, you can see what the domesticated cotton seed looks like. So there's been a lot of phenotypic change. And a question comes up is, as the domestication process happens, does that correlate with an increased amount of genetic load as well? So first, if we look at Gossypium hirsutum and tabulate the number of deleterious mutations in both wild and domesticated accessions, there are more fixed deleterious mutations in the domesticated accessions. And again, we see that D bias over the A subgenome. But the number isn't really all that different. It is more, but it's not really all that impressive. If you look at Gossypium barbadense, we don't have any wild individuals of that in this study. And so the numbers of fixed deleterious alleles in the domesticated is about the same roughly in hirsutum, but you see the trend is flipped where there's more fixed A alleles than D alleles. And finally, if we compare wild and domesticated Gossypium herbaceum, we can see that there's pretty much the same number of fixed deleterious mutations in the wild and domesticated accessions of herbaceum. So this is an interesting trend, but I was also interested in sampling more time points through the domestication process. So both for Gossypium hirsutum and for Gossypium barbadense, through this big resequencing project, we found that there's broadly 2 different land races in both of the domestication processes. And whenever we look at the fixed deleterious mutations in Gossypium hirsutum, we see a huge disparity in mutational load between the two land races. This is still a novel result. We're not exactly sure what's causing this, but one possible explanation is that we know that there's been purposeful introgression from barbadense into hirsutum through the domestication lens. So it could be that the decrease in deleterious snips we see in landrace 2 is actually due to adaptive introgression through the domestication process. And similarly for Gossypium barbadense, we see surprisingly a very similar pattern, although we don't know that there's been as much intentional introgression from hirsutum into barbadense. And so this pattern is a little bit more mysterious and still a little bit less resolved as to what may actually be causing it. So finally, a few takeaway points that I've learned today. The mutational load in polyploids can either be less than, greater than, or equal to diploids. If we don't see an absolute trend, which is kind of surprising and counter to what we would have expected, that polyploids can handle more deleterious load than their diploid progenitors do. But this leads to the question, do polyploids actually accumulate deleterious mutations at a different rate than diploids? And if they do, what does that rate look like? Is it only for recessive deleterious mutations, or is it for incompletely dominant, or partially dominant, or completely dominant, or over dominant? It's just not resolved, and it's a really difficult question to get at without a really large mapping population and a really big sequencing database. Secondly, broadly, we see that there's more deleterious alleles that are fixed in DT than in AT, which is a really interesting phenomenon, and leads to the question, what causes this different amounts of load? Is this a possible stepping stone to bias fractionation in the future, where the DT genome may just accumulate more and more mutational load, and then genes are just lost because they're so deleterious that it's actually beneficial just to get rid of them rather than to actually deal with the load that they carry. And finally, the last observation, maybe there's a founder effect that I've identified in 83 and 87 in Hawaii and the Wake Atoll. We probably, to get better at this, we need to have more sampling of these two species. And it would also be really interesting to know if the deleterious mutations in these two species actually have any effects on the phenotype, and if they've adapted to their environments well enough. So that's all that I have, and I'm happy to take any questions that you guys have.
All right, well, thank you, Justin, for a wonderful talk. As usual, let folks can enter their questions in the chat box, and then if you are brave or interested, feel free to please turn on your video and audio and go ahead and ask that in person. And we'll just organize that there by sort of raising our hand or calling out in the chat box that you have a question.
Let's see if I can.
All right, Robin, you want to go ahead and turn on your video and audio if you want? Feel free to pop in here.
Hi. Yeah, it was a great talk, but I was wondering, I might have missed it, but what do the synonymous SNPs look like when you compare AT and DT? That might help with what the effective population size question you were talking about was.
Yeah, great question. So I didn't look specifically at synonymous sites here, but those sites would have ended up in the very mildly deleterious category. I do have, actually, let me see. So I do know broadly what the, no, I don't have that here, never mind. Yeah, so I don't have the rates for the synonymous sites in the populations, but based on other gene evolutionary rate studies, there's pretty equal evolutionary rates within the polyploid species. So we wouldn't expect that that one subgenome is evolving faster than the other.
Well, no, I would say it's more. So you had the question, could it be just a bottleneck effect? But then you should see the same pattern in synonymous SNPs, because the bottleneck would affect both synonymous and non-synonymous. So I think if you add the comparison, it might answer the question for you.
Yep. Yeah, I completely agree. And something that I'm working on now is actually doing some modeling to see like how many of these SNPs are shared between the different species as well. So if this is a bottleneck in 83, we would expect those sites to be fixed in 83 and only in 83 if it was a founder effect as well. Yeah, that's definitely something that I'm working on next. Yep. Thanks for your question.
All right, looks like the next question is from Boas. If you'd like to go ahead and turn on your audio video and ask, or Justin can take it from there too.
Yes, so Boaz asks, is it possible that different sequencing depth is contributing to the number of variants detected? Are the compared sample sequenced to the same depth? Yes, so in the polyploids, we have the same sequencing depth for both of the subgenomes because they are both sequenced as a single sample. And there's also quality filters for the SNP calling. And I did have a minimum threshold for the read depth to make sure that we weren't getting SNP calls from just 3 or 4 reads. I think on average these were like 30 to 40x coverage. So they were pretty high coverage, and they were definitely deep enough to pick up these fixed sites. Great question. I see Jonathan Wendell has a question. At the level of the organism, one of the nice things about looking at polyploids is that any is identical for both subgenomes because they are in the same nucleus, etc. I guess that's not a question. I think that was probably a response to Robin. Yeah.
I actually have a question since no one else has jumped in there.
Sure.
So we thought about this quite a bit too, and did some comparisons. I think we talked about this last year in Belgium, one of the things I always wonder about, we sort of see similar things, and that it's not really like we didn't really see a clear pattern, but I kind of wonder how much this is due to allo versus autopolyploidy. I feel like some of the first principle intuition that we have comes from thinking about this in an autopolyploid context. But if you think about allos, it's not necessary that each of these alleles is acting in like a tetrasomic fashion the whole time, right? So I'm sort of curious if you could speak about that, or if you have a sense of how much that might be contributing to the sort of pattern not being quite so clear.
Yeah, I think that I personally think that deleterious accumulation is very different in autos versus allos, and that's because there's no recombination between subgenomes in an allopolyploid. So when you don't have that recombination, you can have alleles that can fix within one subgenome and will never be seen by selection because they can be covered by their homolog. Whereas for an autopolyploid, you still have to get above a certain frequency in every chromosome pair before it can be, before it can drift to fixation. So yeah, I think there definitely is a difference, but it's probably based on recombination between the homologs. Now in a system where there's a lot of recombination between the homologs, that's probably somewhere in the middle. But this has just been so understudied so far that I really don't have any evidence to support if there actually is a difference between those different types.
Oh, excellent. Yeah, I was kind of wondering, exactly, if there was a, if in Gossypium it was well understood if there are parts of the genome that behave, that there's a lot of homologous recombination that are identified versus parts that are clearly like on the other end of the extreme, if there was sort of a way to bin those and compare them. That may not be known well enough yet from the pop genome. I don't know. Yeah.
Well, the good thing about cotton is that there's actually a really huge disparity in genome sizes between the two subgenomes. So one subgenome is literally twice as big as the other one. So there's really not that much homologous exchanges happened. There's not been any documented. There is some controversy whether there's been gene conversion between the two, but that still hasn't been thoroughly explored with the baby sequencing data set like this yet.
All right. I think Brittany's got a question there as well. I see that popped in. We're continuing the simultaneous question session here on the chat box.
Brittany's got her.
First off, great talk, Justin. Really enjoyed it. This is totally just piggybacking off of what Jonathan said in the chat about NE being identical for both parental genomes, and I was just wondering, and I realize that this is, you know, a fairly unique case that you see on only a handful of taxa, but if you do have sort of an imbalance between the parental genomes, I'm thinking in Salvinia, there's a pentaploid that appears to have sort of 3 copies of one parental genome and 2 of the other. You know, would you expect to still have, you know, roughly equal NE?
If you'd have equal NE between those 2 subgenomes? Ooh. I actually don't know.
Yeah, I mean, it was an off-the-wall question, so I wasn't like expecting you to, like, have a code answer for that.
I would expect that selection would act differently on those 2 subgenomes because, you know, for a recessive mutation, in the subgenome with, you know, 3 copies, you'd have to have all 3 copies be homozygous before, be homozygous before selection will see it. So those 2 would differ. But as far as how the infected population size varies, I'm not sure about that. That's a good question.
It's a pretty niche thing to come up anyway, so yeah, but just curious. Thanks.
Yeah, no, it's a really interesting system, and there's a lot of, you know, gene dosage balances that would occur in that too. Yeah. Sounds like a fun system though.
We'll see.
Complicated, but
yeah.
fun.
Cool. Thanks.
All right. Well, I think that's all the questions right now for Justin. If anybody has any other questions, feel free to save those and we can ask them at the end as usual. I will also point out that I haven't actually posted a schedule anywhere. I should probably do that. There's a plan in the open, sort of based on some suggestions from other folks that we might do like an open panel sort of discussion session or an open Zoom session to discuss questions like this. So at the end of the fall here. So save these kinds of discussions as well for some fun discussion later this semester. All right, well, with that, I'd like to thank Justin for a great talk, and we'll switch over to Suhir and let her take over the screen. All right. Well, our next speaker is Dr. Suhir Marsid, who is currently a postdoctoral fellow in Christian Landry's lab at the University of Laval. Suhir has an engineering degree in biotechnology from the Institut National. I'm going to butcher the French, and I apologize right away. But the Institut National des Sciences Appliquées et de Technologie of Tunisia in 2010. She moved to Paris for a master's degree in genomics at the University of Versailles, Saint-Quentin and Yvelines. Exactly. I'm going to pronounce these like Spanish and some bad version of English, and I apologize ahead of time. And where she worked on the microbial genomic dynamics in prokaryotes. She then worked on earned her PhD at Montpellier in the field of microbiology and biotechnology. And the main part of her PhD work was supervised by Dr. Sylvie Dequin and Dr. Virginie Galliot with the study of adaptive advantage of horizontal gene transfer in wine yeast. And she's currently studying with Christian Landry on yeast evolutionary genomics, working on interspecific hybrid genome evolution and polyploidy, which she's going to tell us about today. And I'm really excited to hear her talk, which is, I've seen a preview of some of this, and it's really amazing work. So with that, I'm going to let Suhir take it away.
So thank you, Mike. So first, I would like to thank you for organizing this great Polyploidy webinar and for giving me the opportunity to present my work today about. So I will present my work today about the rate of whole genome duplication in hybrids. So as you know, polyploidy or whole genome duplication is an important evolutionary force across the tree of life. So it is an important mechanism of speciation and generally leads to increased biodiversity. So as you know, whole genome duplication is extensively present in plants where it is a common mechanism of speciation. And although more rarely, it occurs also in several animal groups. Also in fungi and also in microorganisms such protists or archaea and bacteria. And in spite of the importance and prevalence of polyploidy, the factors contributing to polyploidization remain not well understood. So a relevant hypothesis proposed in the early 20th century by Winch, so he proposed that hybridization might be the cause of polyploidy. So this concept has since been developed and refined by the most important plant evolutionary biologists, and there is still an ongoing debate about whether or not the extent of parental divergence will drive whole genome duplication. So Winch's hypothesis started to be questioned when Chiara and Ono proposed that there is two types of polyploids: autopolyploids deriving by which chromosome sets comes from the same species, and allopolyploids which derive from hybridization followed by whole genome duplication. So the existence of autopolyploids seems to contradict the fact that hybridization is the only cause of whole genome duplication. However, autopolyploids have been considered for a long time to be maladaptive and very rare in natural populations. And it has been suggested that there is a higher probability of whole genome duplication in hybrids because combining two divergent genomes in the same cells would increase the rate of whole genome duplication. Alternatively, it has been proposed that the positive correlation between parental divergence and allopolyploid speciation could be related to the successful establishment of polyploids rather than their formation. So indeed, hybrids are generally sterile, and whole genome duplication may restore their fertility. Also, allopolyploids may have an increased adaptive potential due to the transgressive segregation that may arise by hybridization. On the contrary, autopolyploids will not benefit from transgressive segregation, and it has been proposed that their fertility will be reduced because of multivalent pairing during meiosis. However, more recent studies showed that autopolyploids are much more prevalent than what we thought, and their reduced fertility has been questioned because it was not always observed. So today, the role of parental divergence in triggering whole genome duplication still under discussion and not well understood. So testing these alternative models will need a direct measurement of whole genome duplication without the confounding effect of natural selection. So in this context, my postdoctoral work aimed to investigate whether hybridization can trigger whole genome duplication without the confounding effect of natural selection. So we proceeded to a direct measurement of whole genome duplication in hybrids and intralineage crosses. So we used yeast as a model. So for that, we constructed 14 different crosses with different levels of parental divergence, from intralineage crosses to interspecific hybrids. We used natural yeast isolates representing 3 incipient species, SPB, SPC, and SPA from the wild Saccharomyces paradoxus yeast, and each sister species, Saccharomyces cerevisiae. So all these crosses have been classified in 4 types according to their parental divergence. So we have VLB, VLC, and VLA or VLS with very low parental divergence, lower than 0.4%. L1 and 2 with low parental divergence, about 2%. N1 and 2 with medium parental divergence, about 4%, and the interspecific hybrid H1 and 2 with high parental divergence, about 15%. So each cross have been done between 64 to 96 independent times. So we obtained a total of 1,352 independent lines. And we also included in our study 152 lines of intralineage crosses described previously by Hal and colleagues. So we ended up with a total of 1,304 independent lines. So these lines have been evolved through repeated bottlenecks by randomly selecting and streaking colonies in plates with solid rich media for reduced selection. So it had been streaked each 3 days for 770 generations. Then we analyzed the ploidy variation at different generation time points of all the 1,304 lines, and we used flow cytometry for that. So the ploidy analysis allowed us to calculate the cumulative number of tetraploids. So we identified a total number of 32 tetraploids. And so we found that whole genome duplication occurred in the 3 types of hybrids and in only one of the intralineage crosses, the VLC cross. So we found that within 770 generations, the rate of whole genome duplication varies among crosses from 0 to 11%. Also, we observed that whole genome duplication within intralineage crosses occurred, is observed only in the 3 independent VLC crosses. So, which suggests that some genotypes are more prone to whole genome duplication. And so because we used the SPC strains in these VLC crosses, we can suggest that these SPC strains are intrinsically more prone to whole genome duplication. So to test this, we analyzed the ploidy of more than 360 wild North American Saccharomyces paradoxus isolates from the 3 different lineage, SPA, SPB, and SPC. And consistent with our observation in the mutation accumulation experiment, SPC is the only lineage from which a natural tetraploid strain was found. Also, we previously showed that the parental SPC haploid strains are more prone to whole genome duplication. So we analyzed the ploidy of more than 180 isolated colonies from the haploid SPC parental stocks. And so here we have haploids, and as you can see, we identified several diploidized cells in the parental stock of haploid SPC strains. So these results confirm that SPC strains are more prone to whole genome duplication. Also, we found that whole genome duplication is observed in L1 and 2 hybrids. So these hybrids share parents with the SPC parents with VLC intralineage crosses, and also share parents with the VLB intralineage crosses where whole genome duplication did not occur. So this suggests that the higher rate of whole genome duplication in L1 and 2 hybrids is potentially a trait inherited from their SPC parental subgenome. We also observed that whole genome duplication occurs in hybrids, but not in all biological replicates. So this suggests that some combination of genotypes are more prone to whole genome duplication. So indeed, for example, for the M1 hybrids, so this hybrid share parents with the VLA and VLB intralineage crosses where whole genome duplication did not occur. And the same thing for H2 hybrids, which share the same parents with VLS and VLB intralineage crosses, and also where whole genome duplication did not occur. So these results suggest that the higher rate of whole genome duplication in M1 and 2 and H2 hybrids is not due to an intrinsic ploidy instability of their parental subgenomes. And so these results provide evidence that whole genome duplication can be triggered by hybridization. Then we tried to investigate the consequences of whole genome duplication on fertility in intralineage crosses and hybrids. So Darlington in 1937, he proposed that there is an inverse relationship between the fertility of a diploid hybrid and to the tetraploid to which it gives rise. So he reasoned that at high parental divergence, at lower parental divergence, the intralineage diploid cross will have a high fertility because the pairing between the homologous chromosomes will occur correctly. However, the corresponding tetraploid will have reduced fertility because the pairing may occur between any pairs of homologous chromosomes, leading to multivalent pairing and uneven segregation. On the contrary, at high parental divergence, the diploid hybrid will be sterile because of the failure of homologous chromosome pairing. And however, the tetraploid to which it gives rise will be fertile because of the correct or consistent bivalent pairing between the identical homologous chromosomes. So to test this hypothesis in our system, we induced meiosis in all 32 lines where whole genome duplication occurred, and we calculated counted the meiotic progeny survival before and after whole genome duplication. So we calculated the percentage of fertility, and in all the crosses, and as you can see, the fertility of diploids decreases with parental divergence. However, after whole genome duplication, the fertility of tetraploids is highly increased in hybrids compared to diploids. And contrary to what Darlington hypothesized, we didn't see a difference between the fertility of tetraploids and diploids in intralineage crosses, VLC crosses. So our results show that whole genome duplication increases the fertility in hybrids and has no negative effect on the fertility of intralineage crosses. Then we tried to investigate why some genotypes or combination of genotypes are more prone to whole genome duplication. So it has been previously proposed that combining two divergent genomes in the same cells would increase the rate of genomic changes and whole genome duplication. Indeed, so hybrids are in general characterized by genomic instability, which is an increased rate of alterations in the genome leading to an increased rate of aneuploidy, variation rates and sizes of loss of heterozygosity, chromosomal rearrangement, or single nucleotide mutation. Also, polyploidy is recognized as one of the typical genomic instability hallmarks in cancer cells. However, little is known about the possible role of genomic instability in increasing the rate of whole genome duplication in hybrids. So we hypothesized that the higher rate of whole genome duplication in some genotypes or combination of genotypes could be due to an overall increased genomic instability in these crosses. So to test this hypothesis, we sequenced the genome of lines from the different three hybrid types and from the intralineage crosses VLC and VLB. So we sequenced 33 to 39 independent diploid lines. Soon after mating and at the end of the experiment. So we ended up with 778 sequenced genomes. In this analysis, we considered diploid lines because we reasoned that crosses with more intense genomic instability would also have more unstable diploid lines. So we then investigated the rate of aneuploidy and loss of heterozygosity, two typical genomic instability hallmarks. So indeed, aneuploidy analysis showed that the VLC intralineage crosses showing the highest rates of whole genome duplication also exhibit the highest aneuploidy rate, followed by also the L crosses or hybrids. Also, the chromosome gain and loss analysis revealed that the chromosome 12 is the most unstable chromosome among VLC intralineage crosses and hybrids. And the high instability of this chromosome could be a testimony of increased genomic instability in the VLC crosses and hybrids. Indeed, so this chromosome in yeast contains the ribosomal DNA array locus, which is a highly repeated sequence of 9 kb encoding for ribosomal DNA. So this region has been described to be one of the most unstable parts of the genome due to the repeated recombination and copy number variation. Then also, we also observed a higher genomic instability hallmarks in the biological replicates in M and H hybrids, which show higher rates of whole genome duplication. So indeed, we analyzed the size of loss of heterozygosity segments among the different crosses, and we found that the H2 hybrids, which show higher rate of whole genome duplication, also have a significantly longer tract of interstitial loss of heterozygosity segments than the H1 hybrids. So the long tract range LOH events have been described to be more frequent under replication stress and have a greater contribution in genomic instability in cancer cells. Also, we observed a difference in the rate of line decline in between M1 and M2 hybrids. So during our mutation accumulation experiment, we observed that there is many lines that went extinct before the end of the experiment. And we observed that the M1 hybrid, which show higher rate of whole genome duplication, also show much higher rate of line decline than M2 hybrids, which suggests that there is a higher genomic instability leading to the frequent segregation of highly deleterious variants compared to the M2 hybrids. So our results overall show that whole genome duplication correlates with higher genomic instability. And finally, we also try to investigate whether whole genome duplication will increase genetic variability in intralineage crosses and hybrids. So for that, we sequenced the genome of all the 32 lines where whole genome duplication occurred soon after mating and at the end of the experiment. And so aneuploidy analysis show that there is indeed a significantly higher aneuploidy rate in tetraploids compared to diploids. Also, we observed different patterns of chromosome gain and loss between diploids and tetraploids. So for example, we observed that there is higher chromosome loss of small chromosomes in diploids, for example, here chromosome 1 and 3. However, in tetraploids, we observed a higher frequency of larger chromosomes. So this suggests that the loss of large chromosome is less deleterious in tetraploids due to the multiple copies present in the cell. So our results confirm that whole genome duplication may increase genetic diversity in the intralineage crosses and also in hybrids. So in conclusion, in this study, we tried to investigate the rate of whole genome duplication in the intralineage crosses and hybrids, and we found that some lineages are more prone to whole genome duplication, and also that whole genome duplication can be triggered by hybridization through the combination of some genotypes. We found that also that there is a higher whole genome duplication rate, the higher whole genome duplication rate correlates with a higher genomic instability. So the genomic instability, whether intrinsic to some genotypes or triggered by hybridization, might accelerate whole genome duplication. And we also found that whole genome duplication leads to increased fertility and genetic diversity that may contribute to the establishment of both autopolyploids and allopolyploids. So to finish, I just would like to thank all people that contributed to this work. So first my supervisor, Christian Landry, and Mathieu Hainaut and Guillaume Charon, which helped me a lot with the mutation accumulation experiment, and Anna Fijarzik with the genome sequencing data. And also I would like to thank all the Landry Lab members for their help and support. And thanks for listening.
All right, well, thank you for a wonderful talk, Suhir. As always, let's have folks type their questions in the chat box. And if you'd like, you could pop on your video and ask a question live. All right, well, I have a real quick question. So, so it seems like do you have a sense of what causes genomic instability to be higher in some genotypes than others? Is it driven by sequence divergence, or yeah, what is known about that? I guess I'm just sort of curious.
Yeah, I think so. Yeah, I think that genetic divergence may play a role in increasing genomic instability. But as you can see in our crosses, some of them is not systematic. So maybe even the ones that don't show higher whole genome duplication, but we see some hallmarks of genomic instability. So maybe there is a threshold of genomic instability that will induce whole genome duplication. That could be incompatibilities between some genes or some stress, like oxidative stress, that may be induced also in hybrids, for example, because of divergence also. In intralineage crosses, that could be a mutation in genes involved in mitotic cell progress or meiotic also cell progress. So that could be also the stress. We don't know exactly. In fact, in hybrids, we observed that there is higher genomic instability, but the molecular processes underlying this are not very well understood. But yeah.
Yeah, that's really interesting. All right, any other questions for Suhir? All right. Well, any questions? Oh, for any other questions for Justin or Suhir, as we all sort of digest after a couple of talks there. All right. Well, I would like to thank both Suhir and Justin for speaking today. I really enjoyed both of these talks. Oh, I see Jonathan says he has a question. All right, I'll slow my shutdown of the. Jonathan, if you want to pop on video there and ask it, that works.
Mute.
There you go.
Video. Hey, I'm here. Hi, everybody. Just a really naive question, pop-gen question, theoretical question. There's been some chat about NE in the sidebar, and I think it's really useful to distinguish NE as it was originally intended as a population level phenomenon. Standing population interbreeding size, right? An organismal level process from all the other ways that NE is now used. So so we started talking about what could be the genetic load in founding individuals or in the founding gametes is related to NE because those founding populations, their load is determined in part by NE. But at some point, let's say you have a monophyletic origin of a polyploid and it starts to evolve. After how many generations will the founding load become absolutely irrelevant? Will equilibrium have been restored such that you can now look at the prediction that load is identical in the two genomes of an allopolyploid without having to be concerned at all about what the progenitor load looked like. So that's kind of that's a numerical question for one of you population geneticists to teach me about. You can do it later by email.
Sorry?
People can send in email responses, or you can start a Twitter chat. Was your question clear?
No, I sorry.
Okay. Well, sorry, Suhir. I'm talking about effective population size, and mostly because of the chat that came up related to Justin's talk, we can set it aside. Everyone probably has more important things to go do right now than listen to me babble. But nice talk, Suhir. Nice talk, Justin.
Thank you.
Absolutely. Well, I was going to say, yes, definitely. Somewhere though, Jonathan, I mean, you know, I'm trying to think through my head here if it's 4 in or how many generations there, depending on whether it's tetrasomic or has gone back to, you know, if it's sort of diplodized or bivalent pairing, until you hit sort of equilibrium at all those neutral loci. But then it's probably going to vary across the genome depending on that. But I would wager that probably many polyploids that are at least a couple 100,000 years old or might be have reached that equilibrium already. I know that the Capsella work, there was a paper a few years ago by from Stephen Wright's group, right, showing the different contributions to the Capsella allotetraploid. You could still see the legacy of the, you know, the deleterious substitution in one of the diploid progenitors. So, but that's a relatively young allopolyploid as far as I remember as well. So
yeah, and that's a pretty wild system too, because one of the diploids is completely outcrossing with a huge any.
Exactly.
The other one I believe is like
the extreme.
Selfing.
It's the extreme example. Yeah, and I think that, you know, the other thing that compounds that too then would be how much gene flow there's been from the diploid progenitors. That makes this all that much messier as to what that actually.
Let's start by keeping it simple.
Yeah, sure.
Let's say there's no gene flow. It's a straightforward allopolyploid. There's no homologous exchange. Nothing untoward. We're simply trying to. What I'm trying to do is get to the point where we now are confident that by looking at an allopolyploid that contains 2 different subgenomes, we have isolated all the external population level and ecological forces from all the internal genetic and molecular forces that can impact accumulation and distribution of genetic load. So that's a nice thing because you have a controlled experiment where both genomes are in the same nucleus, in the same populations, with the same pollinators, blah, blah, blah. So I'm just trying to get to the point where donors have some sense of understanding of how many generations it takes to get to that point.
Yeah, I mean, I think it's in that range of, I want to say 4 in, but I'd have to go back and look myself. Someone else might be more appropriate to answer that.
Well, or is it possible that there's a different equilibrium for the 2 subgenomes? Because once you have standing variation, or once you have a set determined amount of variation, it's hard to change that totally through neutral processes. And it's hard to imagine where, you know, the mutational input should be the same between the 2 subgenomes.
Okay, potentially naive guess here. But if you have an allopolyploid, and we're assuming that it is largely, if not completely, reproductively isolated from its parents, then doesn't that kind of like clean the slate for any, such that both of the parental genomes are now basically an any of 1, because you have one individual that's become this reproductively isolated polyploid?
I think if there's a single polyploidization event, then yes.
Yeah, obviously that.
Whether that's how polyploidy happens generally, or if there's multiple recurring events. I mean, you could have multiple events between two populations where one has really low and one has really high.
Mhm.
So even if you develop a system where there's no gene flow back from the diploids,
Mhm.
there could still be unequal amounts of variation between the subgenomes.
Yeah.
Different load at the start, at the onset.
Right.
Okay.
One unhealthy gamete, one healthy gamete.
Mm.
And I would add, if one parent is also an outcrosser, then you also have this recessive load that now in an allopolyploid you might have to deal with. Which
One
definitely
wavy.
was the case for Swayzega when we examined it.
I suspect when allopolyploids have wildly different genome size, as in cotton, it's not outrageous to think that the two subgenomes have unequal mutational rates, even post-formation, and different recombination frequencies, different recombination is mutagenic, transposable element activities, all that stuff. And that can contribute to load. But that's
Just because
this is where I'm trying to get, because this way you can really study it and you know it's not due to, it's not due to self and route crossing. It's not due to
Right. Well, I think that
population level.
Yeah, I mean, I think the default sort of chimera sort of model sort of thinking about it would be like 4NE basically if it's a neutral allele, right, till it's sort of till you achieve sort of fixation. If you think of it in that way, for those sites, but a lot can happen, and if the parents are really different, then you've got all that sort of homologous recombination. I mean, I think the biggest thing that would screw this up is the multiple origins and homologous recombination. But if you get far enough away from all of that, then 4NE generations is probably enough to assume that things are equaled out. And so for something like these cottons, where I mean they're old, right? They're not they're not spring chickens in the polyploid world. So
Well, and as Brittany pointed out, NE is probably puny.
yeah,
And they're monophyletic. So 4NE happens, could be
pretty
important.
fast. It's absolutely right. It's one of the advantages here, actually. Yeah. So so you get there pretty quickly. Yeah.
Okay, you modelers, help us out.
Well, yeah, no, absolutely. I know. Hopefully, Paul will address some of these things at some point. I'm sure he's hiding right now.
I'm not hiding.
Oh, you're not hiding. Oh, okay.
I'm just waiting for the situation to fully develop, I suppose.
fully develop meaning conclude.
No. Yeah. So I don't know. I mean, I just think I don't know. I'm in a deep dark hole with Brassica napus, and it's just I just don't think that the situation that we're trying to sort of contrive as being very simple exists in reality. And so yeah, it just gets really complicated really fast, to a point where like trying to simplify it, I mean, like we're at the point where like variant calling is extremely difficult because the A and the C genomes are similar enough where you have things that get like mismapped, or like, you basically just have a bunch of SNPs that look like they're at 50% frequency when they're really not, and things that are like at 25% frequency or 75% frequency when they're really not, because there's all of these extra old genome copies as well. And so yeah, trying to like think theoretically about how long it takes for a newly established allopolyploid to reach equilibrium is, yeah, I don't know. It's extremely difficult because it's going to start jettisoning stuff, and yeah, I don't know.
Yeah.
I mean, if we were interested in studying something simple, we wouldn't be working on polyploids.
Yeah, that's true.
Yeah, trying to understand how one genome evolves is hard enough, let alone multiple at the same time.
Absolutely. Yeah. Well, and I was gonna say, Paul, we'll be talking about that in another hour and a half at lab meeting, right?
Yes, we will. So.
All right. Well, yeah. I have to get to a faculty meeting, and actually, let's talk about this. We should save this. I mean, we should save this at the end of the semester. As I said before, we'll have an open sort of forum discussion, and this could be a fun topic for that. I think these are sort of perennial issues that that we have in polyploid research. And so it might be a fun sort of set of questions to talk about deeper there in this kind of open discussion format. That I think it's a really great question. I always default to just saying for any when these things pop up, and I don't know how bad of an answer that is. Hopefully that's somewhere in the middle of a range, but anyway. But it'd be fun to explore that later this fall. And so thank you, Jonathan, for that great question.
Yeah.
And again, thank you to Justin and Suhir for their the great talks today. All right. Well, I'm going to go ahead and close this out because I'm now. I don't know if they'll miss me at faculty meeting, but I probably should go. But I just want to highlight the speakers for the next Polyploid webinar here on September 28th. We'll have Will Booker, who will talk to us about the complex history of whole genome duplication and hybridization in polyploid North American gray tree frogs, as well as Shengchen Shan, who will tell us about transcriptome dynamics of the inflorescence in reciprocally formed allopolyploid Trigopogon misclus in Asteraceae. And with that, I'll see everyone in 2 weeks, and hopefully we can continue some of these discussions over email and save some of these as well for later this semester. I again appreciate Suhir and Justin for 2 wonderful talks today. Thank you.
Thanks, Mike. Thanks, Suhir. Thanks, Justin.